Good afternoon, everyone. Can you hear me? Great. I would like to thank the organizers for inviting me. It's a wonderful place, wonderful weather compared to Boston. So today I would like to present to you a relatively new project, a new direction in my laboratory using sensory stimulation to modify Alzheimer disease. So I want to first just talk about brain oscillations. So we know that in the brain, when large group of neurons, connected neurons actually, fire synchronously together, then they produce oscillatory brain waves at many different frequencies. So this pretty much summarizes all the different frequencies from very slow to the very high frequencies known as the sharp wave ripples, higher than 200 hertz or 200 cycles per second, that can be detected in the brain. And turn out that these different frequencies of neural synchronous oscillations are evolutionarily conserved. You can detect them in very small mammals such as bat and mouse, and you also see them in primates such as human brain. Okay, even the frequencies themselves, you can see they're highly comparable from mouse to humans. So to us and to many people, this suggests that this oscillatory rhythms of neural firing is a very fundamental property of the brain. And you would imagine that it must be very important for the brain function. So there are a lot of hypotheses kind of over the years proposed by people in terms of why is the oscillatory rhythmic neural activity is important for the brain function. But what I think I like the most is really the idea that oscillatory synchronization of neuronal activity can bind neurons in the brain into ensembles which engage in specific computational functions. So we know that cognitive processes depend on coordinated interactions among distributed large-scale brain networks. And the synchronous firing of neurons is very likely to be important for the rapid propagation of information across very long-distance brain regions, and which are necessary to support these various cognitive functions. So I just want to give you an example. For instance, we know very well that the ventral tegmental area, VTA, in the midbrain is very well connected to hippocampal regions and to the cortical region, particularly the prefrontal cortex network. And there are glutamatergic, GABAergic, and dopaminergic projections that go to and from the VTA to these distal brain regions. So I'm giving you an example of a paper published by Yuri Buzaki a few years ago, showing that there are really robust rhythmic oscillations that can be detected across all these different brain regions. And they particularly can detect gamma oscillations between and binding these different regions. And this coherence is particularly strong between the VTA and the prefrontal cortex when the animals are engaged in working memory tasks. So again, suggesting that this very active synchronization and binding of these neurons really is very important in supporting higher order brain functions. And speak of higher order brain functions, these gamma oscillations really have been speculated to be necessary for sensory perception, attention, consciousness, and working memory. So what is gamma oscillations? A simple definition can be seen as activation of local circuits of excitatory and the fast spiking inhibitory neurons that resonate at 30 to 80 hertz. That can give rise to gamma oscillations, and these oscillations can easily be detected by electrophysiological recordings for the local field potential. And very interestingly, I think this gamma oscillation is particularly important for modulating the spiking of the excitatory principal neurons. So as illustrated here, gamma oscillations restrict neuronal spiking at its peak and permits spiking when it decays. So the end product is that you have this extremely sharp spiking patterns of this pyramidal cells. And this sharp spiking is speculated to be important for sensitive high order brain function. And there's also evidence for coupling of different wavelengths of frequencies, such as gamma has been shown to be coupled to, say, theta oscillation, which is a slower wave oscillations, particularly during particular behavioral state. And the interactions can lead to bursts of gamma oscillation critical for spike timing. And the spike timing, I think there's a lot of research activity around it, can serve as internal clock for encoding experience. Okay, so a lot of introduction and oscillations. And I just have one slide that I want to say a few words about Alzheimer's disease. You have heard about Alzheimer's from Eric Kandel's talk this morning already. He told you that it's not normal aging, and it is probably one of the most pressing problem facing the modern society, because it affects many, many people worldwide. 50% of people older than 80 or 85 years old are going to have Alzheimer's disease. And today, there is no cure. There's not even a treatment that can manage or control the disease, let alone prevent the disease. So the disease is really well known for its pathological features, including the extracellular deposition of amyloid plaques, which are made of the small beta amyloid peptides. The most abundant species are A beta 40 and A beta 42 peptides, as well as the very abundant neurofibrillary tangles. These are intracellular aggregation of hyperphosphorylated tau proteins. And in the brain of Alzheimer's patients, they also show severe brain atrophy. This is because of the loss of neurons and loss of synapses. And the brain also has very prominent neuroinflammation. So you may or may not know that turn out gamma oscillations are disrupted in Alzheimer's disease. So, so in humans, this is an example of MEG. You can see compared to age-matched control, the gamma oscillation power is clearly reduced in Alzheimer's patients. And also in mouse models, it has been shown by Leonard Milke and colleagues that in their human mutant APP transgenic mouse model, they found that the fast spiking PAF albumin positive interneurons or GABAergic neurons show impaired synaptic function. And this also is associated with reduced gamma oscillations, where they show around 40 hertz oscillation is greatly reduced. And also this mice show greatly increased epileptic form of neuroactivity. So this is an indication of reduced stabilization of the network, as well as impaired cognitive function. So all of these gamma oscillations in humans and in mouse models were all previously measured in the symptomatic stage of the animals. So that makes it very difficult to discern whether this impaired gamma oscillations is the consequence of the Alzheimer disease pathology or it contributes to the development of Alzheimer disease. So a graduate student, Heina Alcarino, in my lab, she wanted to really look more closely at the role of gamma oscillations in the etiology of Alzheimer's. So she decided to look at much earlier stage of a mouse model. So she used the well-established 5X transgenic mouse model. These animals express 5 different human familial Alzheimer disease mutations in the APP as well as presenilin 1 genes. And this is absolutely a very robust amyloid model. You can see some diffuse plaque deposition already in the 2 months old. And by 6 months old, these mice show cognitive impairment as well as deposition of plaques in the hippocampal formation. And in the earlier stage, you don't see the plaques in the hippocampus. So Hannah decided to evaluate the integrity of gamma in the very young 5XFAD mice around 3 months old. So, so this project was a very fruitful collaboration between my laboratory and the laboratories of Emery Brown, as well as Boyden at MIT. And this is Hannah Alcarino. And she worked closely with Annabelle Singer, who at the time was a postdoctoral fellow in at Boyden's laboratory. So, so really, Annabelle is a very well-established in vivo electrophysiologist. So what she could do is to measure gamma oscillations in a awake behaving mouse. So, so what Annabelle did was to train the mouse to run on a air-supported treadmill through a virtual maze. So then she inserted an electrode into hippocampal CA1 to measure gamma oscillations during running when theta oscillations also occur. And also we could measure gamma when mouse during resting. This is when the Huei ripples occur. Okay, so so I'm gonna just quickly show you the recording results. So these are the raw traces as well as filter traces in the 2 behavioral states. And here you can see the beautiful theta oscillations and the gamma underneath during running. And when we compare the gamma power between the 5XFAD and the wild-type little-mate control, we don't really see a difference. Okay, and then during resting, you can see these Huei ripples, and it is well known that during Huei ripples, gamma oscillations are also enhanced. So here is a power spectral heat map to show you the Huei ripples and the gamma oscillation, and this is an enlarged view of the gamma. And in terms of the instant frequencies during the oscillations, they were of a unimodal distribution centered around 40 Hertz. But very interestingly, during Huei ripples, the gamma oscillations in the 5XFAD mice were clearly impaired compared to the wild-type little-mate. So in a very young 5XFAD mouse model, 3 months old, before the manifestation of behavior deficits, before even the deposition of hippocampal amyloid plaques were detected, these mice already show impaired gamma oscillation. So the question is really, what's the, what does this mean? What does impaired gamma oscillation mean? So then, so it is well established in the literature that the beta amyloid peptides are actually shown to be modulated by neuronal activity. So this has been shown both in vitro model and in vivo models. And I'm just showing you an example from a publication by Dave Holtzman, where they introduce PDX, picrotoxin, to increase neuronal activity, and this results in increased A-beta levels in the interstitial fluid of the brain tissue, and using TTX, tetrodotoxin, to reduce neuronal activity results in reduced A-beta levels. So Hannah decided to look at potential modulation of A beta by gamma oscillations. So first, we need to modulate gamma oscillations. And it turned out that we actually knew how to do that. So through a collaboration with Chris Moore, many years ago now, back in 2009, paper was published, we showed that optogenetically we can drive the PAP albumin positive interneurons in the cortex, and which we can, and upon light illumination of these neurons, we can drive beautiful gamma oscillations. And the increased LFP power really is centered around 40 hertz, very much similar to the kind of gamma we detected in the hippocampus. Okay, so basically we introduced the AAV carrying the channelrhodopsin-2 diol into the cortex of the PV Cree animals. So, the Cree recombinase ensure that the channelrhodopsin-2 is only expressed in the PV neurons. So we used the same strategy. Hannah crossed the PV Cree mice with 5-FAD, and in the 3-month-old mice, we have channelrhodopsin expression in the hippocampus PV neurons, and then we use different conditions to activate these PV positive neurons. So we shine blue light, but at different frequencies, okay? So we have a no stimulation control, and then a 40 hertz stimulation, and then a random stochastic stimulation, average around 40 hertz. So LFP recordings show that only the 40 hertz stimulation, not the random stimulation, results in a sharp increase in gamma oscillations. Although, when you just look at the amount of neuronal spikings, both 40 hertz stimulation and random stimulations induce about the same amount of spiking. We then look at the consequence on beta amyloid peptide levels. So, Hannah, after 1 hour of optogenetic activation of these PV neurons, she then harvested the hippocampi tissue and then performed A beta ELISA to look at amyloid beta 1 to 40 and 1 to 42 levels. And very interestingly, we found just 1 hour of PV neuron activation can have a huge impact on the A beta levels, both 40 and 42. We see about a 40 to 50% reduction of these peptides following 40 hertz stimulation. And this is very frequency specific, because if we use 8 hertz to drive this PV neurons, we don't really see a reduction, and random stimulation actually results in a huge increase in A beta. And also, this is cell type specific. If we drive the KIMK2 positive excitatory neurons, there's no effect, regardless which frequency we use. Okay, so this result were very surprising to us at the time. And so we wanted to know really how gamma oscillations could lead to reduced A beta peptides. So A beta level is known to be controlled by how much A beta is generated in neurons or by neurons, and how much A beta is cleared, which is from the brain, which is primarily carried out by other cell types, such as glial cells. So we first look at A beta production, and A beta is produced from the beta amyloid precursor protein APP, which is a transmembrane protein. APP undergoes sequential proteolytic cleavages by beta secretase and gamma secretase to produce A beta. And beta secretase cleavage results in two products known as the N-terminal soluble APP and C-terminal fragments. Interestingly, we found that 40 hertz optogenetic activation of PAP albumin neurons results in a reduction of both the APP N-terminal and C-terminal fragments. So this result suggests that gamma oscillations actually reduced A beta production by neurons. And this is interesting. But then we really wanted to know whether this is the only mechanism. So, what we did was we harvested, again, the hippocampal tissue from the stimulated animals and performed RNA sequencing. And we found that there are about 500 genes show differential expression following gamma stimulation. Many genes are downregulated, but also there are genes that are upregulated. And among the upregulated genes, we found, very surprisingly, a good 35% are genes that are highly enriched in microglia. And this is surprising because normally microglia constitute about 5% of the total cell composition in the hippocampus and the cortex. So this suggests to us that microglia actually respond very robustly to this neuronal oscillation activity. But I should also point out that many other cell types also show differential gene expression. So among the upregulated genes in microglia, we found that there are genes that are known to be involved in microglia phagocytosis, microglia proliferation, and microglia chemotaxis, which is migration of microglia. So microglia really are a very interesting population of cells in the brain. They are known to be the innate immune cells, but they also play many different roles known to regulate synaptic formation and elimination. But its most well-known role is to serve as the scavengers in the brain, normally surveying the brain environment and trying to get rid of toxic waste such as amyloid or infectious agents. And microglia is also known to exhibit several different states. It can be phagocytic, supposed to be neuroprotective. It can also be inflammatory to further damage the brain tissue. So with that interesting gene expression profile of microglia, we decided to take a look of what the microglia look like. And this is, I would say, the most amazing result I've seen. I continue to be fascinated by how the microglia look following stimulation of gamma. So, here we use IBA1 to label microglia in the hippocampus, and I think you can just see yourself. This is what microglia look like in the 5-FAD mice before the stimulation and after the stimulation. Okay, they look completely different. And in particular, I want to point out the cell size, the cell body size of microglia is increased by about 100%. So the cell body just like a puff up. And simultaneously, the processes seem to retract a little bit. So the length of the process is shorter, okay? And also, when we do the co-immunostaining of microglia with A-beta, we found that, so you can see that A-beta is much reduced. But anyway, a lot of these microglia show A-beta inside their cell body. So this suggests to us that this microglia become extremely active in taking up this microglia, in this A-beta, engulfing this A-beta. So I think this plays a very important role in the phenotype we observe, which is this dramatically reduced A-beta levels following gamma stimulation. So just to summarize a little bit, I show you that just 1 hour of gamma frequency stimulation that can produce two biological processes that may contribute to reduce A-beta. One is that neurons produce less A-beta, and the other is that these microglia are really strongly recruited and engaged, and they undergo dramatic transformation in terms of their morphology and function, and they become very active in phagocytosing A-beta. So while these results are very interesting, a lot of my colleagues, especially colleagues who are physician scientists, pointed out that and optogenetics is really invasive. So if you really want to have any chance of trying this approach out in humans, you have to use a much less invasive method.